Welcome, friend. Find yourself a comfortable place to rest. Let your body settle into stillness as we begin a gentle journey together, one that will carry you softly towards sleep while unveiling one of nature's most profound secrets. Tonight, we're going to explore the hidden mind of the green world, the intelligence that exists in every leaf, every root, every branch reaching toward the sky. And if you find value in these explorations, in this space we share, I invite you to subscribe and become part of this community of curious minds seeking rest in wonder. Take a breath now, slow and deep. Let it out. We begin. What if I told you that the silent forest surrounding you, the houseplant on your windowsill, the grass beneath your feet, all possess a form of intelligence? Not the intelligence we typically imagine, not thoughts racing through neural pathways or consciousness flickering behind eyes, but something else entirely. Something we're only now beginning to understand. For centuries, we've looked at plants as the passive backdrop to the drama of animal life, as living things certainly, but living in the way furniture might live if only it could photosynthesize. We've been so very wrong. Imagine a forest just before dawn. The darkness hasn't quite lifted, and in that pre-light stillness, nothing seems to be happening. No deer are moving between the trees. No birds have begun their morning songs. The forest appears frozen, dormant, waiting. But beneath this apparent stillness, an entire world of activity unfolds. In the soil, root tips are actively exploring, questing through the dark earth like fingers reading braille, sensing the chemical signatures of nutrients and water, of neighboring roots both friend and foe. Above ground, leaves are already preparing for the sun's arrival, having tracked the passage of night through their internal clocks, their cellular machinery ramping up in anticipation of the first photons of dawn. Trees are communicating through vast underground fungal networks, sharing resources, sending warnings, making what can only be described as decisions about where to send precious sugars, which offspring to support, how to respond to threats. The forest is not waiting. The forest is thinking, in its own distributed, ancient way. This revelation represents one of the most dramatic shifts in biological understanding in recent decades. We're witnessing what some scientists call a revolution in botany, a complete reframing of what it means to be a plant. The journey to this understanding has been long and often contentious. In the Victorian era, Charles Darwin himself conducted experiments on plant movement and sensitivity, observations that were largely dismissed by his contemporaries who couldn't imagine that plants might be more than mere automata. In the early 20th century, brave researchers who suggested plants might have something akin to a nervous system were ridiculed, their careers threatened. The very term plant neurobiology, coined in the early 2000s, sparked fierce debates that continue today. How dare we apply terms like intelligence and behavior and decision-making to organisms without brains? But here's what we've learned, what decades of careful research have revealed. Intelligence doesn't require a brain. Not the way we thought it did. The definition we now work with, proposed by scientists who study cognition across all domains of life, is this: intelligence is the capacity for adaptive beneficial behavior, the ability to sense the environment, process that information, and respond in ways that increase survival and reproduction. By this measure, plants are not just intelligent. They're remarkably so. They assess their surroundings with an array of senses that rivals and in some ways exceeds our own. They estimate how much energy they'll need for particular goals and then pursue the optimum path to achieve those goals. They take measures to control certain environmental resources. They perceive themselves and can distinguish between self and non-self, between their own roots and those of their neighbors. They process information and modify their behavior accordingly. All of this without a single neuron. Consider what this means. A plant doesn't have a central command center, no brain sending orders to distant limbs. Instead, it operates as a distributed intelligence, a democracy of cells where decisions emerge from countless local interactions. Each root tip, for instance, contains a region called the transition zone, an area packed with cells engaged in intense electrical and chemical signaling. Some researchers argue these root tips function analogously to neural networks, processing sensory information and directing growth responses. Whether or not we accept this controversial comparison, the capability is undeniable. Roots make decisions. They choose which direction to grow based on gradients of moisture, nutrients, and oxygen. They avoid toxic substances. They recognize obstacles and grow around them. They even appear to engage in strategic competition, altering their growth patterns in response to the proximity of other roots, deploying more resources to zones where competition is fiercest. The seed that decides when to germinate faces one of life's most critical choices with precious little information. Germinate too early, and the emerging seedling dies in winter's cold. Wait too long, and you're crowded out by competitors who claim the best spots in the sun. The seed integrates cues about temperature, moisture, light quality, the length of cold periods it has experienced, even chemical signals from other plants in the environment. Somehow, in a structure containing only 3,000 or 4,000 cells with no central coordinator, this information gets processed and a decision emerges. Germinate now. Or wait. It's a strategic calculation that determines whether this individual plant will live or die, reproduce or vanish from the gene pool. That's not simple mechanics. That's something far more sophisticated. And plants face countless such decisions throughout their lives. Where to send limited resources, whether to invest in growth or defense, when to flower, how many seeds to produce, which fruits to abort when resources run short. These aren't predetermined programs running without variation. Plants adjust their strategies based on their individual circumstances, their history, what they've learned from past experiences. Yes, learned. Because plants have memory too, a capability we once thought impossible without neurons, but we're getting ahead of ourselves. First, we need to understand how plants know what's happening around them in the first place. We humans experience the world through our five senses, or at least that's what we learned in elementary school. Vision, hearing, touch, taste, smell. We're quite proud of these senses, these windows into reality. But plants, it turns out, may perceive the world in more ways than we do. They lack eyes, yet they see. They have no ears, yet they hear. They possess no nose, no tongue, yet they detect chemicals with an acuity that puts our senses to shame. They feel touch, respond to gravity, sense electric fields, detect sound waves, and measure the passage of time with extraordinary precision. Some scientists suggest plants may have 15 or more distinct sensory systems, each tuned to a different aspect of their environment. Let's begin with light, since for a plant, light is everything. Light is food, light is information, light is life itself. You might think that plants simply detect whether it's light or dark, perhaps measure intensity, but the reality is far more nuanced. Plants perceive light through several families of photoreceptor proteins, each responsive to different wavelengths, each providing different types of information. They don't see images the way our eyes do, it's true. No plant is forming a picture of the world in anything resembling our visual experience. But what they can do is remarkable. Through proteins called phytochromes, plants detect red and far-red light, wavelengths that tell them not just about the sun's position but about their neighbors. You see, when sunlight passes through leaves or reflects off them, the ratio of red to far-red light changes. Leaves absorb most red light for photosynthesis but reflect far-red. So when a plant detects a shift toward more far-red light, it knows, in its plantish way, that it's being shaded or that competitors are growing nearby. This triggers what's called a shade avoidance response. The plant elongates its stem, reaching upward, trying to overtop its rivals in the race for sunlight. It's a competitive behavior, a response to sensing other plants in the environment, and it's mediated entirely through light perception. Cryptochromes and phototropins, other families of photoreceptors, respond to blue light. These proteins help plants determine the direction light is coming from, enabling phototropism, the growth toward light that we observe when a houseplant leans toward a window. But blue light perception does more than guide growth direction. It influences stomatal opening, those tiny pores on leaf surfaces that let carbon dioxide in and water vapor out. It affects the plant's circadian rhythm, that internal clock that anticipates the daily cycle of day and night. It even plays a role in photoperiodism, the ability of plants to measure the length of day and night, information crucial for timing flowering and other seasonal events. Plants know what season it is not because someone shows them a calendar, but because they've been counting. They measure how long the nights are, how many hours of darkness they experience. Short-day plants flower when nights grow long in autumn. Long-day plants flower when nights are brief in summer. Day-neutral plants ignore photoperiod altogether, using other cues. But for many species, this light-based timekeeping is essential. A plant that flowers at the wrong time misses its pollinators, wastes its reproductive effort, leaves no offspring behind. So plants measure time through light, tracking both the daily cycle and the yearly progression of seasons, planning their life history accordingly. Perhaps most mysteriously, there's the case of boquila space trifoliata, a climbing vine native to the temperate rainforests of Chile and Argentina. This plant does something that has left scientists genuinely puzzled. It mimics the leaves of whatever tree it climbs, changing the shape, size, color, and even vein patterns of its own leaves to match its host. A single vine climbing through different trees will display different leaf forms on different branches, each matching the local host. The mimicry is so precise that even botanists have difficulty distinguishing boquila leaves from those of the host plant. The obvious assumption was that the vine achieved this through some chemical signal received upon physical contact with the host. But then researchers did an experiment that shocked the botanical community. They placed boquila near plastic artificial plants, no chemical signals possible, and the vine mimicked the plastic leaves. The implication is unsettling. Can plants somehow see the shape of leaves around them? Do they possess some form of vision at a distance we don't yet understand? The mechanism remains unknown, but the behavior is undeniable and thoroughly documented. It suggests that our understanding of how plants perceive their world may still be in its infancy. So plants see light in ways that inform them about energy availability, direction, time of day, season of the year, and the presence of competitors. But light is only the beginning of their sensory world. Plants also hear, though again not in the way we typically think of hearing. Sound, after all, is vibration traveling through matter, and plants are exquisitely sensitive to vibration. The first hints of this came from experiments on plant growth. Researchers noticed that playing certain sounds or music near plants sometimes affected their growth rate, their germination, even their resistance to disease. For years, these results were dismissed as pseudoscience, lumped in with claims that plants respond to human emotions or that talking sweetly to your petunias helps them thrive. And indeed, some of those claims were nonsense, wishful thinking mixed with poor experimental design. But the careful work persisted, and what emerged was a genuine phenomenon. Plants do respond to vibrations, but not to music per se. They respond to sounds that matter to them in an ecological sense, sounds that carry information relevant to their survival. Root tips, for instance, can detect the sound of water flowing through pipes, even when those pipes are buried in soil and not leaking. The roots grow toward the sound, using the vibration as a cue to locate water sources. In experiments where water pipes were buried near plant roots, the plants detected and grew preferentially toward the pipes even when no moisture was present in the surrounding soil. They were homing in on the acoustic signal, the faint vibrations of water moving through the pipe. This behavior makes perfect evolutionary sense. For a plant, Finding water is life or death. A sensory system that can detect the sound of underground streams or water moving through soil after a rain provides a significant advantage. Above ground, plants detect the vibrations of insects. When a caterpillar chews on a leaf, it creates a specific pattern of vibration, different from the vibrations caused by wind or rain or even from the touch of a human hand. Some plants have been shown to distinguish between these different vibrations, responding with defensive measures only to the specific frequency pattern of insect feeding. In one elegant experiment, researchers recorded the sound of caterpillars eating leaves, then played those recordings back to plants through speakers that vibrated the leaves without any actual herbivory. The plants responded by increasing production of defensive chemicals, the same response they'd show to actual caterpillar attack. They heard the sound of being eaten and prepared their defenses. Even more intriguing, flowers can hear pollinators. Research on evening primrose flowers revealed that within minutes of detecting the buzz of a bee's wings, the flowers increased the sugar concentration in their nectar. Not by much, but measurably, enough that it could make a difference in whether a bee chooses to visit or moves onto a competing flower. The flowers themselves appear to act as the sensory organ, the bowl-shaped petals functioning somewhat like an ear, focusing sound vibrations. When researchers covered flowers with glass jars that blocked sound but allowed light and scent to pass, the flowers no longer increased nectar sweetness in response to bee recordings. But flowers covered in mesh, which allowed sound but not bees themselves, still responded. The plants were definitely detecting the acoustic signal. This acoustic world extends even to plant-to-plant communication. Stressed plants, particularly those suffering from drought or mechanical damage, emit ultrasonic clicks and pops, sounds far above the range of human hearing but potentially detectable by other plants. Research from 2023 demonstrated that tomato and tobacco plants produce these sounds when water-stressed or when their stems are cut. The mechanism appears to be cavitation, the formation and collapse of air bubbles in the plant's water transport system, a bit like the creaking of a tree limb but at ultrasonic frequencies. Whether other plants detect and respond to these sounds remains an open question, but it's a tantalizing possibility. Imagine a forest where trees can hear each other's stress, where the sound of drought in one area alerts plants in adjacent areas to prepare for water scarcity. And then there's touch, perhaps the most obvious of plant senses but no less remarkable for being familiar. We've all seen time-lapse videos of climbing plants circling and reaching, tendrils wrapping around supports, bin plants spiraling up poles. This growth is directed by touch. The side of the tendril that makes contact with the support stops growing or slows its growth while the opposite side continues, causing the tendril to curl around the object. Darwin studied this in climbing plants, fascinated by how they found and grasped supports. What he documented was THIGMOTROPISM, directional growth in response to touch. But touch does more than guide climbing. It affects overall plant development in a phenomenon called thigmomorphogenesis. Plants that experience frequent touch or wind grow differently from plants raised in still air. They become shorter, stockier, with thicker stems and more compact growth. It's why plants grown indoors often look leggy and weak, why greenhouse plants sometimes struggle when transplanted outside, and why vegetation on windswept coastlines or mountain ridges grows stunted and gnarled. The mechanical stimulus of wind and touch triggers changes in gene expression that alter the entire growth strategy of the plant. Simply brushing a plant regularly, even gently, is enough to trigger these changes. The plant remembers being touched, and it responds by building a body better suited to a physically challenging environment. The most dramatic touch responses are rapid movements, behaviors so quick they seem almost animal-like. The Venus flytrap, that marvel of botanical engineering, can snap shut in 1/10 of a second, fast enough to trap a fly. The trigger is touch, specifically the bending of tiny hairs on the inside of the trap leaves. But here's what makes it truly remarkable: the plant counts. A single touch isn't enough. The trap won't close unless at least 2 hairs are bent within roughly 20 seconds, or the same hair is bent twice in that time frame. This prevents the plant from wasting energy closing on raindrops or wind-blown debris. It's a basic form of short-term memory and arithmetic, implemented without neurons through electrical signaling and mechanical changes in cell shape. When the threshold is reached, an action potential, an electrical signal similar in principle to those in animal nerves, races through the trap, triggering the rapid release of elastic energy stored in the curved trap surfaces. The 2 lobes snap together, the marginal spines interlock, and the insect is caught. MIMOSA space PUDICA, the so-called sensitive plant, is another master of rapid movement. Touch a leaf, and it folds closed in seconds. The entire plant can respond within minutes, leaves all along the stem folding up, sometimes even the whole plant drooping as if it suddenly wilted. The mechanism involves rapid water movement out of specialized cells at the base of each leaflet, a loss of turgor pressure that causes the leaf to collapse. But why? The traditional explanation is defense, that folding makes the plant look less like a tasty meal or exposes thorns on the stem. More recent thinking suggests it might startle herbivorous insects, literally shaking them off when the leaves move suddenly. Whatever the reason, the behavior is unmistakable and utterly unlike our stereotypes of plant life. Finally, consider chemical perception, the plant equivalents of smell and taste. Plants exist in a world of chemical signals, constantly sampling their environment through receptors on root hairs, leaf surfaces, and flower parts. They detect nutrients in soil, sensing nitrogen, phosphorus, potassium, and micronutrients with remarkable specificity. Different forms of nitrogen, nitrate versus ammonium, trigger different growth responses. Localized patches of phosphorus cause roots to proliferate just in that area, mining the nutrient concentration. This isn't passive diffusion and uptake. It's active foraging guided by chemical sensing. Plants also detect volatile organic compounds released by their neighbors. When one plant is attacked by herbivores, it releases a complex bouquet of VOCs, and nearby plants detect these compounds and activate their own defensive systems preemptively. It's a neighborhood watch system, chemical screams of being eaten that prepare others for potential attack. The receiving plants don't just respond to any volatile chemicals. They discriminate, responding to specific blends that signal genuine threats while ignoring background volatiles. Some species can even recognize chemical signatures from close relatives versus strangers, altering their growth patterns to compete less aggressively with kin. Below ground, the chemical communication becomes even more intricate. Roots release exudates, complex mixtures of sugars, organic acids, amino acids, and secondary compounds that shape the microbial community in the surrounding soil, recruit beneficial fungi, repel parasitic plants, and potentially signal to neighboring roots. Some plants release allelopathic chemicals that inhibit the germination or growth of competing species, a kind of chemical warfare in the struggle for space and resources. Other plants release chemicals that mobilize nutrients, making insoluble minerals available for uptake. And in what may be the most sophisticated chemical signaling of all, plants communicate through shared fungal networks, sending messages encoded in sugar concentrations, nutrient transfers, and signaling molecules that travel along fungal hyphae connecting root to root across the forest floor. This world of sensation, this rich perceptual environment that plants inhabit, is fundamentally different from ours yet no less real. We see the green world as silent, passive, unaware. But in truth, it's constantly sensing, constantly processing information from light and sound and touch and chemistry, constantly making adjustments in response to a world that's far from static. The question that naturally arises is this: what happens to all that sensory information? Is it simply triggering automatic responses, stimulus and reaction with nothing in between? Or is there something more, some processing, some integration, something we might dare call cognition? That's where memory and learning enter the picture, capabilities that fundamentally change how we think about plant minds. And it's there, in the realm of memory, that we continue our exploration, letting ourselves drift deeper into understanding, deeper into the restful wonder of botanical consciousness. Scientists once believed memory required a brain, required neurons and synapses where experience could be encoded in the strengthening or weakening of connections between nerve cells. Without such machinery, how could any organism remember? This assumption seemed so fundamental, so obviously true, that for decades researchers didn't even look for memory in plants. Why would they? Plants don't have neurons. They don't have synapses. Therefore, they can't remember. Except they can. And the experiments that proved it are as elegant as they are surprising. Let's travel back to 2014, to a laboratory at the University of Western Australia where a researcher named MONICA space GAGLIANO was working with MIMOSA space PUDICA, that sensitive plant that folds its leaves when touched. Gagliano knew that mimosa's rapid folding response costs energy. The plant has to pump water out of cells, then pump it back in to re-inflate the leaves. If the stimulus is a genuine threat, worth responding to, fine. But if it's harmless, repeatedly folding would be wasteful. Could the plant learn to ignore a harmless stimulus? Could it habituate? Gagliano designed a simple but clever apparatus. Individual mimosa plants were placed in pots, and these pots were dropped from a height of about 6 inches onto a cushioned surface, enough to trigger the folding response but not enough to damage the plant. Then they were dropped again, and again, and again, every 5 to 6 seconds. At first, every drop caused the leaves to fold. But after several drops, something changed. The plants stopped responding. Their leaves stayed open. They had learned, or habituated, to the repeated harmless stimulus. Now, habituation is a basic form of learning seen in many animals. Snails habituate. Sea slugs habituate. It's considered one of the simplest forms of learning. But in plants? This was new. But Gagliano went further. She tested whether the plants remembered this learned response. She left them alone for a day, then tested them again. The plants still didn't respond to drops, even though a full day had passed. They remembered. She waited a week. Still habituated. She couldn't quite believe it herself, so she waited a month. And after a month, when she started dropping the plants again, they still didn't fold their leaves. 1 month of memory. In bees, in insects that have actual brains, long-term memory is usually defined as lasting 24 hours. Mimosa, with no brain at all, remembered for at least 30 times longer. But the experiment had another layer. Gagliano tested whether this was truly learning or just fatigue, just the plant being too tired to respond anymore. She shook the habituated plants, a different stimulus, and they folded immediately. Not tired then. They had specifically learned that the dropping was harmless while remaining responsive to other stimuli. And she tested different contexts. Plants habituated to drops under low light conditions didn't retain the habituation when tested in bright light, and vice versa. The learning was context dependent, more sophisticated than simple fatigue. These plants were integrating information about the stimulus with information about environmental conditions, storing this as some kind of memory trace, and retrieving it appropriately. The biological basis for this memory, as far as we understand it, involves changes at the cellular and molecular level. Plants don't have neurons, but they do have electrical signaling, and they do have calcium-based signaling networks very similar to those involved in animal memory. When a plant is stimulated, calcium ions flood into cells, and the pattern of calcium concentration changes across time and space appears to encode information. Repeated exposure to the same stimulus changes how cells respond to that calcium signal, essentially adjusting sensitivity. Additionally, plants use epigenetic modifications, chemical tags attached to DNA that alter gene expression without changing the DNA sequence itself. These modifications can persist for extended periods, providing a molecular substrate for memory. The story of plant memory extends beyond habituation. There's memory involved in vernalization, the process by which many plants require a period of cold before they can flower. Winter wheat needs to experience weeks of cold temperatures, recording this experience and only becoming competent to flower after the cold requirement is met. The molecular mechanism involves a protein called VRN2, which maintains a repression of flowering genes through the cold period. The longer the cold exposure, the stronger the repression, and only after sufficient cold does the plant release this repression and allow flowering when warm weather returns. The plant remembers winter. This memory can last months, carrying information from the cold season into the warmth of spring. Even more striking, plants remember stress and use that memory to prepare for future stress. In experiments with tomato plants, researchers exposed plants to drought, then re-watered them and let them recover. Weeks later, up to 5 weeks in some studies, the same plants were subjected to drought again. And these previously stressed plants handled the second drought better than control plants that had never experienced water shortage. They closed their stomata more quickly to conserve water, adjusted their metabolism more efficiently, and showed less damage overall. They remembered being drought-stressed and had essentially trained themselves to handle it better next time. This is adaptive memory, memory that improves fitness, and it's implemented through both physiological changes and epigenetic marks that persist after the stress is removed. Then there's the controversial work on associative learning, experiments that stretch the boundary of what we're willing to accept plants can do. Gagliano, the same researcher behind the mimosa experiments, published a study in 2016 that claimed plants could learn associations between neutral and meaningful stimuli, a form of Pavlovian or classical conditioning. She worked with pea plants, which naturally grow toward light, a behavior called phototropism. She placed pea seedlings at the base of a Y-shaped maze. At the end of one arm was a light, and alongside that light was a fan producing airflow. At the end of the other arm, nothing. The plants, unsurprisingly, grew toward the arm with the light. Then came the training phase. Some plants were grown in mazes where the light and fan were always together in the same arm. Other plants had the light and fan in opposite arms. After this training, Gagliano tested the plants in a new maze with only the fan, no light. And here's where it gets interesting. Plants that had previously experienced light and fan together grew toward the fan, as if expecting light to be there. Plants that had experienced light and fan in different locations showed no such preference. The interpretation: plants trained with light and fan together learned to associate the neutral stimulus, airflow, with the meaningful stimulus, light, and then changed their behavior based on that learned association. The scientific community met this claim with deep skepticism. Replication attempts have had mixed results, with some labs unable to reproduce the findings and others seeing partial effects. The debates continue, arguments about methodology, about sample size, about what counts as genuine learning versus statistical noise or experimental artifact. What's clear is that even raising the question, even attempting such experiments, was unthinkable a generation ago. Now it's a matter of serious investigation. Whether or not pea plants learn Pavlovian associations, the fact that it's a genuine scientific debate represents a massive shift in how we think about plants. What makes all of this memory possible, what allows plants to store and retrieve information about past events, is their electrical and chemical signaling systems. And it's to those systems, those internal communication networks, that we now turn our attention, drifting further into the architecture of plant intelligence, into the mechanisms that allow a distributed organism to behave as an integrated whole. When a Venus flytrap catches a fly, when mimosa leaves fold in response to touch, when an electrical signal races from a wounded leaf to distant parts of a plant triggering defensive responses, what's actually happening at a biophysical level is remarkably similar to what happens in animal nerves. Remarkably, but not identically. The similarities and differences are both instructive. In 1873, a British physiologist named John Burdon-Sanderson, working in collaboration with Charles Darwin, made the first recording of an electrical signal in a plant. The plant was Dionaea the Venus flytrap, and what Burdon-Sanderson measured was an action potential, a rapid change in electrical voltage across cell membranes, the same type of signal that neurons use to communicate in animal nervous systems. This was a startling discovery at the time, suggesting that plants and animals, despite their vast differences, might use similar electrical strategies for rapid signaling. An action potential is fundamentally a wave of change in electrical charge that propagates along a cell membrane or from cell to cell through connected tissue. In resting conditions, living cells maintain an electrical voltage difference across their outer membrane, the inside typically negative relative to the outside. This voltage, called the membrane potential, is maintained by pumps and channels that control the movement of ions, charged particles like potassium, sodium, chloride, and calcium. When a cell is stimulated strongly enough to reach a threshold, channels in the membrane open, ions rush across, and the membrane voltage flips, becoming temporarily positive inside. This depolarization triggers adjacent membrane areas to depolarize in turn, creating a wave of electrical change that travels along the cell or through connected cells. In animal neurons, this process happens very fast, milliseconds, and the signal can travel at speeds up to 110 meters per second in myelinated nerve fibers. In plants, the process is somewhat slower, but still impressively rapid for an organism without nerves. Plant action potentials travel through vascular tissue, particularly the phloem, those tube-like cells that transport sugars and other compounds throughout the plant. The electrical signal propagates at speeds around 1 meter per second in some species, fast enough to coordinate responses across the entire plant body within seconds to minutes. The ionic basis of plant action potentials differs from animals in interesting ways. In animal neurons, the primary ions involved are sodium rushing in during depolarization and potassium flowing out during repolarization. Plants don't use much sodium. Instead, they rely on calcium influx to depolarize the membrane and potassium and chloride efflux to repolarize. The result is functionally similar, an all-or-nothing electrical signal that propagates without loss of amplitude, but the molecular machinery is different, a case of convergent evolution arriving at similar solutions through different biochemical paths. Plants generate several types of electrical signals, not just action potentials. Variation potentials, also called slow-wave potentials, are induced by mechanical damage or rapid changes in pressure within the plant's water transport system. Unlike action potentials, variation potentials change in amplitude and speed as they propagate, often becoming weaker with distance. They're associated with wounding, with the physical pressure changes that occur when a stem is cut or crushed. The signal spreads through the plant's hydraulic system, a wave of pressure that triggers electrical responses in cells along the way. System potentials are a third type, involving hyperpolarization rather than depolarization, the membrane becoming more negative rather than less. These signals appear to coordinate growth and development across regions of the plant, though they're less well understood than action potentials or variation potentials. All these signals serve to rapidly transmit information across distances within the plant body. When a leaf is attacked by an insect, electrical signals spread from the site of damage to other leaves, triggering the production of defensive chemicals even in tissues that haven't been touched. When roots encounter a pocket of nutrients, signals travel upward, potentially influencing how the plant allocates resources between root and shoot growth. The Venus flytrap's famous snap relies entirely on electrical signals, those trigger hairs detecting touch and generating action potentials that, if they sum to threshold within the 20-second window, cause a massive coordinated change in cell pressure that flips the trap shut. What's especially intriguing is that plants use some of the same signaling molecules that animals use as neurotransmitters, glutamate, which functions as a major excitatory neurotransmitter in mammalian brains, also plays a signaling role in plants. When a plant is wounded, glutamate concentrations increase, and this glutamate can trigger calcium waves that propagate through the plant, ultimately leading to the production of defensive compounds. GABA, gamma-aminobutyric acid, another important neurotransmitter in animals, is found in plants and appears to be involved in stress responses and in modulating electrical signaling. The presence of these molecules in both kingdoms is likely due to shared evolutionary ancestry. Plants and animals diverged over 1 billion years ago from common single-celled ancestors, but some of the basic biochemical toolkits for cellular signaling have been retained across that vast span of time, modified and elaborated in different directions but still recognizably related. This raises a profound question. If plants use electrical signals, if they use an EUROTRANSMITTER-like molecules, if they process information and coordinate responses across their distributed bodies, do they have something analogous to a nervous system? Most scientists would say no, not in any meaningful sense. Nervous systems as we define them involve specialized cells, neurons, that are dedicated to signaling and nothing else. Plants don't have neurons. Their electrical signals travel through cells that also perform other functions, photosynthesis or transport or storage. There's no specialized wiring, no biological circuitry designed exclusively for information processing. Yet some researchers, those working in the controversial field of plant neurobiology, argue that this is a semantic distinction without a real difference. They point to root tips, which contain densely packed cells engaged in heavy electrical activity and which integrate sensory information from multiple sources to direct growth. They point to the PHLOEM, which serves as a long-distance signaling pathway analogous to the way nerves connect different body regions. They argue that we shouldn't require plants to have the same anatomical structures as animals before we acknowledge that they perform similar functions. Function, not structure, should be the criterion. By that standard, plants do have something we might call a decentralized nervous system, a network for rapid information transmission and integration, implemented in a completely different architecture but serving fundamentally similar purposes. The debate is more than academic. It touches on deep questions about what we mean by intelligence, by cognition, by awareness. If we define these terms so narrowly that only animals with brains can qualify, we blind ourselves to other forms of information processing that may be equally sophisticated. But if we define them so broadly that any responsive system qualifies, we dilute the terms into meaninglessness. Finding the right conceptual framework, the right language to describe what plants do, remains one of the central challenges in this field. What's not debatable is that plants live in a world of signals, electrical and chemical, rapid and slow, local and systemic, and that these signals allow coordination and integration that gives the plant functional unity despite its distributed, modular construction. A tree isn't a colony of independent cells happening to live together. It's an individual, a single organism that maintains its integrity through constant communication between parts. And much of that communication, especially when responding to rapid changes or acute stress, happens through electrical signaling that would have astonished botanists of earlier eras. As we drift further into this exploration, as our breathing slows and the boundary between waking thought and dream becomes softer, let's carry this image with us: electrical signals racing through vascular bundles, calcium waves propagating from cell to cell, a green body that thinks, in its way, without a brain but not without a mind. The plant intelligence we're uncovering isn't alien, not completely. It uses some of the same molecular tools we do, faces some of the same problems we do, solves them through information processing and adaptive behavior. And yet it does all this while rooted in place, unable to flee, unable to hunt, forced to deal with whatever circumstances arise exactly where the seed happened to germinate. Perhaps that makes plant intelligence not inferior to animal intelligence but simply different, optimized for a life of stillness in a world of change. And there's more to explore still, more ways that plants coordinate and communicate, not just within their own bodies but with each other, across distances, forming networks that span entire ecosystems. But that journey continues in the next part of our exploration, as we rest deeper, breathe slower, and open ourselves to the interconnected wonder of the natural world. Picture yourself standing in an old-growth forest in early spring. You're surrounded by towering Douglas firs and paper birches, their crowns intertwining far overhead. At your feet, the forest floor is carpeted with moss and last autumn's decomposing leaves. To all appearances, each tree stands alone, an individual competing for light, water, and soil nutrients. This is how we've long thought of forests, as collections of independent organisms engaged in relentless competition. But beneath your feet, hidden in the dark soil, a different story unfolds. The roots of these trees don't merely coexist. They're connected, linked by thread-like fungal networks that stretch for miles, creating what scientists have begun calling the Wood Wide Web, an underground internet through which trees and plants share resources, exchange information, and support one another in ways we're only beginning to fathom. This revelation emerged gradually, over decades of patient research by scientists who were willing to look beneath the obvious, to ask what mycorrhizal fungi, those symbiotic partners that have colonized plant roots for perhaps 400 million years, might be doing beyond their known role in nutrient exchange. The word mycorrhiza comes from Greek roots meaning fungus and root, and for most of the 20th century, the relationship was understood in fairly simple terms. The fungus, living in or around the plant's roots, extends the root system's reach, those fine fungal threads called hyphae exploring the soil far more extensively than roots alone ever could. The fungus breaks down minerals and makes them available to the plant, particularly phosphorus, which is often locked in insoluble compounds that plant roots can't access directly. In exchange, the plant provides the fungus with sugars produced through phosphonthresis, carbon compounds the fungus can't make for itself. A fair trade, mutualism in its textbook form. But here's what researchers discovered when they started carefully tracing these fungal networks. A single fungal individual doesn't associate with just one plant. The hyphae branch and spread, sometimes extending yards or even miles through the soil, and as they spread, they encounter the roots of other plants. And they colonize those roots too. The result is that multiple plants, often of different species, find themselves sharing the same fungal partner, all plugged into the same network. These common mycorrhizal networks effectively connect the plants to each other, creating an underground web of biological connections. Once you realize the networks exist, the next question becomes obvious. Do things travel through them? Can substances move from one plant to another via the shared fungal highway? The answer, demonstrated through elegant radioactive tracer experiments, is yes. Carbon moves. Nitrogen moves. Water moves. Phosphorus moves. A plant with access to abundant resources can send some of those resources through the network to a plant that's struggling. Even more intriguingly, the pattern of sharing isn't random. Research by Suzanne Space Simard, a forest ecologist at the University of British Columbia who pioneered much of this work, revealed that in mixed stands of Douglas fir and paper birch, the two species engage in a complex seasonal exchange of carbon. In summer, when the deciduous birch is fully leafed and photosynthesizing vigorously, it sends carbon to the evergreen fir. In autumn and winter, when birch has dropped its leaves and fir is still photosynthesizing, the flow reverses. The two species are supporting each other, evening out the differences in their seasonal strategies. Even more remarkably, older, larger trees, often called mother trees, appear to play a special role in these networks. They're hubs, superconnected nodes with extensive fungal associations linking them to many other trees. And mother trees don't just absorb resources. They distribute them, sometimes preferentially to their own offspring, seedlings growing in their vicinity that share their genes. SIMARD''s work showed that mother trees recognize kin through chemical signals and send more carbon to related seedlings than to unrelated neighbors. It's a form of parental investment, mediated through fungal networks, that challenges our assumptions about plants as purely competitive and self-interested. But do these networks carry more than just nutrients? Can they transmit information? This is where the science becomes both exciting and contentious. Some studies suggest that when a plant is attacked by insects or infected by pathogens, it can send warning signals through the mycorrhizal network, causing neighboring plants to preemptively activate their defensive systems even though they haven't been attacked yet. The mechanism appears to involve changes in the types and concentrations of molecules being sent through the network, chemical messages that the receiving plants interpret as danger signals. One study that garnered significant attention involved broad beans and aphids. Researchers allowed aphids to infest some bean plants while leaving others uninfested. The plants were connected by mycorrhizal networks. What they found was that the uninfested plants, the ones connected to infested plants via the fungal network, showed increased resistance when aphids were subsequently introduced to them. Their leaves contained higher concentrations of defensive chemicals even before any aphids had touched them. They had somehow been warned. Control plants, not connected to infested plants, showed no such preparation. The inference was that information about the aphid attack traveled through the mycorrhizal network. However, this interpretation isn't universally accepted. Some scientists argue that what's traveling through the network might simply be a change in sugar or nutrient concentrations, a consequence of the infested plant's stressed state, and that the receiving plants are responding to this metabolic change rather than to any specific warning signal. The debate illustrates a recurring challenge in plant communication research, distinguishing between true information transfer, where specific messages are encoded and decoded, versus mere physiological responses to environmental changes. It's a subtle distinction but an important one. Regardless of the mechanism, the existence of these networks and the movement of substances through them has profound implications. It means that forests aren't just collections of individual trees. They're integrated systems where the success of individuals is tied to the health of the community. A forest with intact mycorrhizal networks is more resilient, better able to withstand drought, better able to recover from disturbance. The networks redistribute resources from areas of abundance to areas of scarcity, evening out the inevitable patchiness of soil nutrients and water. They connect generations, allowing the accumulated resources of established trees to support the growth of seedlings in the dim understory. And they may constitute a form of ecological memory, the fungal networks themselves persisting for years or decades, maintaining connections even as individual plants come and go. There's a darker side to these networks too, or perhaps just a more realistic acknowledgement of their complexity. Not all interactions mediated by mycorrhizae are cooperative. Some plants appear to parasitize the networks, taking more than they give, drawing resources from neighbors without reciprocating. The balance between mutualism and exploitation varies depending on environmental conditions, the species involved, and even the individual genotypes of the fungal partners. Some mycorrhizal fungi are more equitable partners than others. The networks aren't a utopian sharing economy. They're sites of cooperation and competition, of reciprocity and theft, of relationships that shift depending on circumstance. And mycorrhizal networks aren't the only way plants communicate through the soil. Roots themselves directly interact, detecting and responding to neighboring roots through chemical signals. Plants release exudates from their roots, complex cocktails of organic compounds, and these exudates can be sensed by other plants. Some species use exudates to suppress competitors, releasing compounds that inhibit germination or growth of potential rivals. Others use exudates to signal identity, allowing the plant to recognize whether a neighboring root belongs to itself, to a close relative, or to an unrelated stranger. Studies have shown that plants grow their roots more aggressively, more competitively, when they detect strangers nearby compared to when they're growing next to kin. Family matters, even underground. The ability to distinguish self from non-self through root-exuded chemicals is itself a sophisticated feat. It requires that the plant have some representation of its own chemical signature, some template against which to compare the signatures of others. This is a form of self-awareness, not consciousness as we experience it, but a biological self-knowledge that allows the plant to orient itself socially in its underground neighborhood. And the consequences are real. Plants that can recognize and cooperate with kin have advantages in some environments, particularly stable environments where relatives are likely to remain neighbors over long periods. The genes for cooperation spread not because they benefit the individual in isolation, but because they benefit the family group, a case of kin selection operating in the plant kingdom just as it does among animals. Above ground, the communication networks are no less intricate, though they operate through an entirely different medium, air. Plants speak in chemistry, releasing volatile organic compounds, VOCs, that drift on the breeze and can be detected by other plants at considerable distances. This airborne chemical communication has been documented since the early 1980s, but only in recent decades have researchers begun to appreciate its sophistication and ubiquity. The classic scenario goes like this. A caterpillar lands on a leaf and begins to chew. Within minutes, the damaged plant begins releasing a specific blend of VOCs. The exact composition of this blend depends on the plant species, the type of damage, even the specific herbivore doing the eating. These aren't random emissions. They're information-rich signals. Neighboring plants detect these VOCs through receptors on their leaf surfaces and in their stomata. And upon detecting them, the neighboring plants activate their own defensive responses, increasing production of chemicals that make their leaves less palatable or less nutritious to herbivores. They're preparing for an attack that hasn't happened yet, based on chemical intelligence received from a plant under siege. But the communication is even more sophisticated than simple alarm signaling. Some plants, when attacked, release VOCs that specifically attract the predators or parasites of their attackers. A lima bean plant infested with spider mites releases a particular blend of volatiles that attracts predatory mites, which feed on spider mites. The plant is calling for backup, recruiting mercenaries to help fight its battle. Corn plants attacked by caterpillars release VOCs that attract parasitic wasps. These wasps lay their eggs inside the caterpillars, and the wasp larvae consume the caterpillar from the inside out. From the plant's perspective, this is an excellent outcome. The herbivore is neutralized, and the plant doesn't have to invest its own resources in chemical defenses. This strategy, called indirect defense, has been documented in dozens of plant species across multiple plant families. It appears to be an ancient and widespread adaptation, and it requires remarkable chemical specificity. The plant must produce the right blend of VOCs to attract the right predators. Produce the wrong blend, and you might attract the wrong insects, or no insects at all. The precision of this chemical signaling, the lock and key fit between plant VOC blends and predator olfactory receptors, testifies to millions of years of coevolution, predators and plants mutually shaping each other's biochemistry. Plants also use VOCs to communicate with each other about more than just herbivore attacks. Some evidence suggests plants can signal about pathogen infections, about drought stress, about nutrient availability. The language of plant volatiles is rich and complex, more nuanced than a simple on-off alarm system. Different blends encode different meanings, and plants can apparently distinguish among these different messages, responding appropriately to each. There's even speculation, though this remains highly controversial, that plants might use VOCs for competitive signaling, releasing compounds that serve as honest signals of their own vigor and size, allowing neighboring plants to assess the competition and adjust their growth strategies accordingly. In dense plant communities, such signaling could reduce wasteful overgrowth, each plant adjusting its investment in height versus root growth or reproduction based on chemical information about its neighbors. Whether plants actually engage in this sort of strategic signaling remains an open question, but the possibility illustrates how much we still don't understand about plant communication. One of the most hotly debated topics in plant communication is the role of biophotons, extremely weak light emissions that all living organisms produce as a byproduct of metabolic processes. These light emissions are far too faint to see with the naked eye, but they can be detected with sensitive instruments. Some researchers have proposed that plants might use biophotons as a communication channel, that variations in these light emissions could carry information detectable by neighboring plants. The evidence for this is intriguing but preliminary. In experiments where plants were exposed to biophotons from other plants, changes in gene expression have been observed in the exposed plants, suggesting some sort of response to the light signal. If this is real, and it's a big if, it would represent a fundamentally different communication channel from chemical or electrical signaling. Light-based communication would be fast, traveling at the speed of light, and directional, potentially allowing more targeted signaling than diffuse chemical messages. But the mechanism by which plants would detect these ultra-weak light signals remains mysterious, and many scientists remain skeptical. This is definitely a frontier area of research, where speculation currently outpaces solid evidence. What's clear beyond debate is that plants don't exist in isolation. They're embedded in webs of communication, below ground through fungal networks and root exudates, above ground through volatile chemicals and possibly through light. These networks connect individuals into communities, communities into ecosystems. Information flows through these networks, information about threats, about resources, about the state of neighbors near and far. And plants respond to this information, adjusting their growth, their defense, their resource allocation in light of what they learn about their world. The integration of multiple communication channels is where things get really interesting. A plant receiving information through its roots via the mycorrhizal network, through its leaves via airborne VOCs, through its internal tissues via electrical signals, all simultaneously, must somehow integrate all these inputs and arrive at a coordinated response. How does a decentralized organism without a brain achieve this integration? We don't fully understand, but it likely involves the same sorts of mechanisms we've discussed before: calcium signaling, electrical potentials, hormone cascades, all intersecting and modulating each other to produce a coherent behavioral output. Different communication channels have different properties, different strengths and weaknesses. Electrical signals are fast but limited to within the plant body or at most to immediately adjacent connected plants. Chemical signals through the soil are slower but can reach farther and persist longer. Airborne VOCs are faster than soil chemicals but more subject to disruption by wind and dilution with distance. MYCORRHIZAL networks provide direct plant-to-plant connections but only to those plants sharing the same fungal partners. By using multiple channels in parallel, plants can achieve both speed and reliability, both local and long-distance communication. And the context matters. In dense forest environments, where MYCORRHIZAL networks are well established and where plants are packed closely together, underground communication through fungal networks may dominate. In more open environments, where plants are spaced farther apart and fungal networks are less extensive, airborne VOCs may play a larger role. Plants adapt their communication strategies to their environments, emphasizing the channels that work best in their particular circumstances. As we rest here, breathing softly, letting this information settle into the quiet spaces of almost sleep, consider what it means that beneath every forest, every meadow, every garden, these conversations are happening. Plants talking to fungi talking to plants. Chemical words drifting on the wind. Electrical whispers racing through PHLOEM. The green world, which appears so silent, is actually humming with communication, a constant exchange of information that we, in our animal-centric worldview, have only recently learned to recognize. And these communication networks enable something even more remarkable, something we'll explore as we drift deeper still, the capacity for plants to make decisions, to choose among options, to solve problems. Because with perception and with memory and with communication comes the possibility of choice, of genuine behavioral flexibility. And that brings us to perhaps the most challenging question of all: do plants make decisions? Can an organism without a brain engage in anything we'd recognize as decision-making? The answer, as we're about to discover, depends on what we mean by decision. And the exploration of plant decision-making takes us to the very roots, if you'll excuse the expression, of what we mean by intelligence itself. In the darkness of soil, a root tip encounters an obstacle. Perhaps it's a rock, perhaps a compacted layer of clay, perhaps another root. The tip pauses, then changes direction, growing around the obstacle. On the surface, this seems like a simple mechanical response, growth redirected by physical pressure. But look closer. Before the root commits to a new direction, it probes, sending out short exploratory branches in different directions, testing the environment. Some of these branches find the path blocked. Others find the way open. The root then invests its growth resources primarily in the directions that showed promise, abandoning the dead-end explorations. This is foraging behavior, search and evaluation followed by resource allocation based on what was learned. and it requires making a choice among alternatives based on information. That's decision-making. Or consider this scenario. A seed sits in the soil, dormant but alive, waiting. Its internal chemistry detects temperature, moisture, day length, the presence of certain chemicals in the soil. All of this information must be integrated. Germinate now, and the seedling might freeze. Wait too long, and competitors will have claimed the best spots in the sun. The seed doesn't have days or weeks to ponder this choice. The window of optimal germination timing might be quite narrow. And yet, somehow, the seed makes the right call often enough that the species persists. How? The mechanism involves an interplay of hormones. ABS-ciacic acid inhibits germination, keeping the seed dormant. Gibberellin promotes germination. The two hormones are antagonistic, each suppressing the other's production. Environmental cues shift the balance. Sufficient cold exposure promotes gibberellin production and suppresses ABS-ciacic acid. Adequate moisture does likewise. Light of the right quality tips the balance toward gibberellin. The seed essentially integrates multiple inputs by allowing them to affect this hormonal balance, and when gibberellin levels rise high enough relative to ABS-ciacic acid, germination proceeds. It's a decision made through biochemistry, a choice emerging from the interactions of molecular signals rather than from neurons and action potentials, but a genuine assessment of options nonetheless. And it's not a deterministic process. Seeds of the same species, experiencing the same environmental conditions, don't all germinate at once. Some germinate immediately. Others wait. This variation is itself adaptive, a form of bet hedging. By spreading out germination timing, even within a single population, plants ensure that at least some offspring will hit favorable conditions even if the timing turns out to be wrong for others. It's a strategic response to uncertainty, a plant version of not putting all your eggs in one basket. And it requires that individual seeds make slightly different decisions despite having nearly identical genetic programs and experiencing nearly identical environments. The variability appears to arise from small stochastic differences in the molecular machinery, noise that gets amplified into different outcomes, randomness harnessed for strategic purposes. Once a plant has germinated and begun growing, it faces a constant stream of decisions about resource allocation. Roots versus shoots, how much to invest in each. Defense versus growth, which to prioritize. Reproduction versus continued vegetative expansion, when to make that transition? These are complex optimization problems, multidimensional trade-offs where the right answer depends on environmental conditions, the plant's current state, its developmental stage, and what its neighbors are doing. Take the decision of when to flower. For many plants, this is the most consequential choice they make. Flower too early, before you've accumulated sufficient resources, and you'll produce few, weak seeds. Flower too late, and you might miss the window when pollinators are active, or you might get hit by frost before seeds mature. The optimal flowering time is a moving target, depending on temperature patterns, day length, the plant's nutritional status, even whether herbivores have been attacking and forcing the plant to divert resources to defense rather than growth. Plants integrate all this information through a complex genetic network involving dozens of genes and multiple environmental sensing systems. The most famous example comes from work on Arabidopsis, a small weedy plant that's become a model organism for plant biology. Arabidopsis measures day length through photoreceptors, measures temperature through still unclear sensors, assesses its nutritional state through internal hormone signals, and integrates all this through a network of genes that either promote or repress flowering. The gene network acts as a computational device, processing multiple inputs and producing a binary output, flower now or don't. The decision that emerges is sensitive to context, appropriate to conditions, and represents a genuine evaluation of alternatives. But perhaps the most impressive examples of plant decision-making come from root foraging, the strategies roots use to find and acquire nutrients in heterogeneous soil. Soil is patchy, nutrients distributed unevenly in both space and time. A root system must somehow search this three-dimensional space efficiently, finding and exploiting nutrient-rich patches while not wasting resources exploring barren zones. Roots do this through a combination of local response and systemic control. When a root tip encounters a pocket of high nutrient concentration, say a patch of high phosphorus, the local response is to proliferate. The root branches, producing many lateral roots just in that area, essentially mining the patch. At the same time, systemic signals, hormones traveling through the plant, inform other parts of the root system about the discovery. These distant roots may then reduce their exploration effort, since the plant's phosphorus needs are being met. This is coordination without central control, different parts of the root system adjusting their behavior based on both local information about what they personally are encountering and systemic information about the plant's overall nutritional state. The specificity of these responses is remarkable. Different nutrients trigger different architectural changes. Nitrate availability stimulates root elongation, helping the plant chase this mobile nutrient deeper into the soil. Ammonium availability stimulates lateral root branching, helping the plant exploit this less mobile nutrient more thoroughly in the zone where it's encountered. Phosphorus, which is extremely immobile in most soils, triggers both lateral root proliferation and increased root hair density, maximizing the root surface area in the patch. The root system is tailoring its morphology to the specific characteristics of different nutrients, exhibiting what can only be called sophisticated foraging strategies. And roots don't just respond to nutrients. They respond to water gradients, growing preferentially toward wetter soil. They respond to oxygen gradients, avoiding waterlogged anaerobic zones. They respond to gravity, generally growing downward, though lateral roots partially suppress this gravitropic response to allow horizontal exploration. They respond to obstacles growing around them. They respond to neighboring roots, both their own and those of other plants, adjusting their growth patterns based on the perceived level of competition. All of these responses must be integrated, and the integration happens without a brain, through the summed effect of local and systemic signals shaping growth at millions of root tips simultaneously. Research comparing root foraging in different species reveals that there's no single optimal strategy. Some species are conservative foragers, growing slowly and steadily, not responding dramatically to local nutrient patches but maintaining a well-distributed root system that can take advantage of wherever nutrients happen to appear. Other species are opportunistic foragers, responding strongly to nutrient patches, proliferating rapidly wherever resources are found, but potentially missing opportunities elsewhere. The optimal strategy depends on the predictability of the environment, the plant's life history, the intensity of competition. Natural selection has tailored each species' foraging behavior to its typical ecological niche. Even within a single species, individual plants adjust their foraging strategies based on experience. A plant that has recently encountered nutrient-rich patches becomes a more aggressive forager, more responsive to subsequent patches. A plant growing in consistently poor soil eventually reduces its foraging effort, conserving resources rather than wasting them on fruitless search. This is adaptive behavioral flexibility, learning from the past and adjusting future behavior accordingly, and it operates through the same sorts of memory mechanisms we discussed earlier: epigenetic modifications, persistent changes in hormone sensitivity, alterations in gene expression that last for weeks or months. Another striking example of plant decision-making comes from the phenomenon of selective fruit abortion. Many plants produce more flowers and initiate more fruits than they can possibly mature. As the fruits develop, the plant must decide which ones to continue supporting and which ones to abort. This isn't random. Plants selectively abort fruits that are smaller, that were pollinated late, that are positioned disadvantageously on the plant, or that developed in locations where resources are scarcer. The result is that the plant concentrates its limited resources on the subset of fruits most likely to produce successful offspring. It's a cost-benefit calculation, an optimization problem solved through selective resource allocation. The mechanism appears to involve competition among developing fruits for hormonal and nutritional signals from the parent plant. Fruits that are developing well, that are receiving adequate resources, produce hormones that signal their healthy state. Fruits that are struggling fail to produce these signals. The parent plant reads these signals and adjusts resource flow accordingly, eventually cutting off resources to the weakest fruits. The decision of which fruits to support emerges from this chemical dialogue between mother plant and developing offspring, a conversation in hormones and sugars that results in strategic abortion. Even PHOTOSYNTHESIS, The most fundamental of plant processes involves ongoing optimization decisions. Stomata, the pores on leaf surfaces, must open to let carbon dioxide in for photosynthesis, but closing them conserves water by reducing transpiration. The plant faces a constant trade-off: open stomata for maximum photosynthesis but risk water loss, or close stomata to conserve water but reduce carbon intake. The optimal stomatal aperture changes throughout the day as temperature, humidity, and light levels change, and it changes over longer time scales as soil moisture changes. Plants continuously adjust stomatal opening, and these adjustments represent ongoing decisions balancing the costs and benefits of water loss versus carbon gain. The sophistication of these stomatal control systems is impressive. They respond not just to current conditions but anticipate future conditions based on time of day and past experience. They coordinate across the entire leaf surface, with stomata in different parts of the leaf adjusting their opening in coordinated waves. They integrate information about light levels, humidity, internal water status, and carbon dioxide concentrations inside the leaf. The control system is distributed, each stomatal guard cell capable of independent response, yet the aggregate behavior shows coordination that serves the plant's interests. So do plants make decisions? If by decision-making we mean consciously weighing options and deliberately choosing, then no, plants don't decide in that sense. But if we mean assessing environmental information, evaluating alternatives, and selecting behaviors that serve the organism's fitness, then absolutely yes, plants make decisions constantly. They're just doing it through different mechanisms than we use, through biochemical networks and distributed sensing and hormone gradients rather than through neurons and brains. The key insight is that decision-making, as a functional capability, doesn't require any particular substrate. It requires information gathering, some form of memory or representation of past states, some mechanism for evaluating alternatives based on likely outcomes, and some way to implement the chosen action. Plants have all of these. Their mechanisms are different from ours, but the functional capabilities are recognizably similar. They solve the same problems we do: how to find food, how to avoid danger, how to reproduce successfully, just using plant-specific solutions. As we rest here, breathing slowly and deeply, letting sleep approach like a gentle tide, we can appreciate the strangeness and the beauty of what we've uncovered. Decision-making without a brain. Intelligence without neurons. Problems solved through chemistry and physics and information encoded in the shapes and sizes of growing organs. The plant world operates by rules foreign to our animal intuitions, yet solving fundamentally similar challenges. And recognizing this, recognizing the intelligence in forms of life so different from ourselves, expands our understanding not just of plants but of what intelligence itself means, of the many possible ways that evolution can craft solutions to the problems of survival. There's wonder in that realization, peace in seeing ourselves as part of a larger community of solving, striving, intelligent life. And as that peace deepens, as our breathing slows further, we can carry these thoughts with us into the realm of dreams, into the restorative darkness where our own minds process and integrate the marvels we've learned. And if this journey into the quiet minds of plants has brought you a little peace, a little wonder before sleep, you are warmly invited to subscribe and return whenever you wish, to continue exploring the gentle edges of science and philosophy as night gathers around you. For now, let your body grow heavy, your thoughts grow light, and your awareness dissolve slowly into rest, carried on the green intelligence of a living world. The sensory sophistication we've explored, the memory systems that store experience, the electrical networks that coordinate responses, the underground connections that link plant to plant, all of these would be meaningless without one final capability: decision-making. Plants must take the information they gather and use it to make choices about where to grow, when to reproduce, how to allocate limited resources among competing demands, and how to respond to threats and opportunities. These are genuine decisions in the sense that matters for survival, selecting among alternative courses of action based on evaluation of the probable outcomes. And nowhere is the reality of plant decision-making more apparent than in the behavior of roots as they forage for nutrients in the complex, heterogeneous environment of soil. Imagine yourself as a plant root, extending through darkness, navigating a three-dimensional maze of soil particles, rocks, air spaces, and the roots of countless other plants. Nutrients are distributed unevenly, concentrated in some patches and nearly absent in others. Water availability shifts with depth and lateral position. Some areas of soil contain toxic aluminum at concentrations that would damage your tissues. You have no eyes, no map, no advanced knowledge of what lies ahead. Yet you must find the resources your plant needs to survive, and you must do so more efficiently than the competing roots of neighboring plants, because in the fierce underground competition for nutrients, failure means starvation and death. This is the challenge every plant root faces, and the strategies they've evolved to meet it are sophisticated and varied. Roots don't simply grow randomly in all directions. They actively forage, adjusting their growth patterns based on what they encounter, proliferating in nutrient-rich patches and reducing growth in nutrient-poor areas, changing their architecture in response to local conditions while also considering the nutritional status of the plant as a whole. This is active decision-making distributed across thousands or millions of root tips, each responding to local cues but coordinated through signals from the rest of the plant. The behavior of roots in heterogeneous soil has been studied extensively, and the findings reveal complex, adaptive responses that vary depending on which nutrients are available and where. Consider nitrogen, which plants need in large quantities for building proteins and nucleic acids. Nitrogen comes in different chemical forms, primarily as nitrate and ammonium. These two forms have different effects on root architecture. When ammonium is locally available, it stimulates the branching of lateral roots, causing roots to proliferate in the ammonium-rich zone. When nitrate is locally available, It doesn't increase branching so much as it stimulates the elongation of existing lateral roots, allowing them to explore a larger volume of soil. These differential responses make sense given the mobility of these nutrients in soil. Ammonium binds to soil particles and doesn't move much, so intensive branching to exploit a localized patch is effective, while nitrate is mobile in soil water, so elongating roots to follow the nutrient as it moves makes more sense. Phosphorus presents a different challenge. It's essential for energy metabolism and nucleic acids, but it's also one of the least mobile nutrients in soil, forming insoluble compounds with aluminum, iron, and calcium that make it largely unavailable to plants. When phosphorus is scarce, plants respond with a suite of architectural and physiological changes. They increase the length and density of root hairs, those microscopic extensions of root surface cells that vastly expand the area in contact with soil. They alter the angle at which lateral roots grow, making the root system shallower because phosphorus tends to accumulate near the soil surface. They release organic acids and enzymes from root tips that can help dissolve phosphorus from insoluble compounds. And they invest heavily in mycorrhizal associations because fungal hyphae are particularly effective at accessing phosphorus from small pores in soil that roots can't penetrate. What's remarkable is that these responses aren't just triggered by nutrient deficiency in the plant as a whole, though that certainly plays a role. Roots also respond to local nutrient availability, detecting nutrients in their immediate vicinity and adjusting their growth in that specific location even if other parts of the root system are encountering different conditions. This requires integration of both local signals, what this particular root tip is sensing right now, and systemic signals, what the plant's overall nutritional status is. The interplay between local and systemic control allows plants to fine-tune their foraging strategy, proliferating roots in rich patches when the plant is generally well-fed but redistributing growth more evenly when the plant is nutrient-starved and needs to maximize exploration. The sophistication of these decisions becomes especially clear in experiments where plants are given choices. In split-root experiments, where a single plant's root system is divided between containers with different nutrient regimes, researchers can observe how plants allocate growth between the two environments. The results show strategic, non-random allocation. When one container has nitrogen and the other has phosphorus, plants balance their root growth between the two, ensuring access to both essential nutrients. When one container has high nutrients and the other has low nutrients, plants preferentially grow roots into the high nutrient environment, but they don't abandon the low nutrient side entirely, maintaining some presence there perhaps as insurance or to monitor for changing conditions. Even more intriguingly, root foraging behavior can be influenced by the presence of competitors. When roots encounter the roots of neighboring plants, their growth patterns change, sometimes becoming more exploratory, sometimes more intensive depending on species and circumstances. Plants can distinguish their own roots from those of others, and some can even distinguish roots of relatives from roots of unrelated individuals of the same species. Kin recognition in plants, demonstrated in several species, suggests that plants adjust their competitive behavior based on genetic relatedness, growing less aggressively when surrounded by relatives, a strategy that makes evolutionary sense if it increases the overall success of family members carrying similar genes. The decisions roots make about where and how to grow have consequences that ripple through the entire plant. A root system that efficiently locates and exploits nutrient-rich patches will support more vigorous growth above ground, more leaves capturing more sunlight, more energy for reproduction. A root system that makes poor decisions, growing into depleted zones or failing to respond appropriately to local conditions, condemns its plant to stunted growth and reduced fitness. The stakes are high, and natural selection has honed these decision-making capabilities over hundreds of millions of years. But root foraging is just one arena where plants make decisions. Consider the germination dilemma, perhaps the most critical decision a plant makes in its entire life. A seed, sitting dormant in soil, must decide when to germinate. If it germinates too early, in fall or early winter, the seedling will likely be killed by frost. If it waits too long, germinating in late spring or summer, it may face competition from other plants that germinated earlier, shading it out or depleting soil moisture. The optimal germination time depends on temperature, moisture, light, and the specific ecological context of the species, and the seed must make this decision based on limited information, primarily what it can sense about temperature, moisture, light quality, and chemical cues in the soil. The decision is mediated by two antagonistic hormones, gibberellin, which promotes germination, and abscisic acid, which inhibits it. The balance between these hormones shifts in response to environmental cues. Sufficient moisture increases gibberellin and decreases abscisic acid. The right temperature range has similar effects. Light of certain wavelengths can tip the balance, but it's not a simple threshold response where germination happens as soon as conditions cross some fixed line. Seeds integrate information over time, and many species require specific sequences of environmental signals. Some need a period of cold followed by warmth, ensuring they've experienced winter before germinating in spring. Others need scarification, physical damage to the seed coat that might indicate the seed has been through an animal's gut or subjected to fire, both of which might create favorable conditions for germination in certain ecosystems. The germination decision is particularly fascinating because it's made by what amounts to a few thousand cells with no central coordinating structure. There's no brain, no CEO cell that receives all the information and issues the germination order. Instead, the decision emerges from interactions among different cell populations in the seed, particularly cells in the radicle, the embryonic root that will emerge first. These cells sense their environment, produce and respond to hormones, and collectively transition from dormancy to active growth when conditions are favorable. It's distributed decision-making at its most stark, a life or death choice made by a tiny community of cells through purely local interactions and chemical signals. Once a plant has successfully germinated and begun growing, it faces a continuous series of decisions about resource allocation. A plant has finite energy and materials available from photosynthesis and nutrient uptake. How should it divide these resources among competing demands? Growth versus defense is a classic trade-off. Investing in physical structures like tough leaves or chemical defenses like toxins reduces herbivory but diverts resources away from growth, potentially causing the plant to be outcompeted by faster-growing neighbors. Investing heavily in growth makes the plant more competitive for light, but leaves it vulnerable to herbivores and pathogens. The optimal strategy depends on the environment. In safe, resource-rich conditions, rapid growth is favored, while in dangerous conditions with high herbivore pressure, investment in defense pays off. Above-ground versus below-ground allocation presents another trade-off. Roots access water and nutrients, shoots capture light. The optimal allocation between them depends on which resource is most limiting. In dry or nutrient-poor soils, more investment in roots makes sense. In deeply shaded conditions where light is limiting but water and nutrients are abundant, more investment in shoots allows the plant to reach toward available light. Plants adjust this balance continuously based on the resources they're encountering, and different species have different strategies even in the same environment, reflecting their particular evolutionary histories and ecological niches. Perhaps most dramatically, plants make cost-benefit decisions about reproduction itself. Flowering and producing seeds is expensive, requiring substantial energy and nutrients. For many plants, particularly annuals, reproduction is a one-time event followed by death, making the timing and scale of reproduction critical. The plant must be large enough and have sufficient resources to produce viable seeds, but waiting too long increases the risk of death before reproduction and may mean missing the optimal window for seed dispersal or pollination. Perennials face related decisions about how much to invest in reproduction each year versus continuing vegetative growth. When resources become limited during reproduction, many plants selectively abort some of their developing fruits or seeds, a process called selective abortion. This isn't random death but active culling, with the plant apparently assessing which fruits are best positioned, largest, or most likely to mature successfully, and allocating resources preferentially to those while allowing others to abort. Studies have shown that plants don't just passively fail to support weak fruits but actively dismantle them, recovering nutrients for reallocation. This is cost-benefit analysis in action, with plants making strategic choices about which offspring to invest in based on the likelihood of success. The timing of flowering represents another complex decision. Many plants use day length as a primary cue, measuring the duration of light and dark periods with remarkable precision to determine the season. This photoperiodic response allows plants to flower when pollinators are active, when weather conditions favor seed development, or when other aspects of their life cycle synchronize with environmental conditions. But day length isn't the only factor. Temperature history matters too, particularly for plants that require vernalization. Some plants integrate information about day length, temperature, nutrient availability, water stress, and even the presence of pollinators, using all these cues to decide the optimal moment to transition from vegetative growth to reproduction. The mechanisms underlying flowering time decisions involve sophisticated epigenetic regulation. Genes that promote or suppress flowering are dynamically regulated through methylation, chromatin remodeling, and other modifications that allow past environmental conditions to influence current gene expression. This creates a form of memory that persists for weeks or months, allowing the plant to make flowering decisions based on accumulated evidence about seasonal progression rather than just the immediate environment. It's a slow form of computation, information processing that operates over developmental timescales, but it's computation nonetheless, solving a complex optimization problem with multiple variables and trade-offs. Even the moment-to-moment regulation of photosynthesis involves decision-making of sorts. Stomata, the microscopic pores in leaves that allow carbon dioxide to enter for photosynthesis, also allow water vapor to escape. Opening stomata increases carbon dioxide availability and thus photosynthetic rate, but it also increases water loss through transpiration. Closing stomata conserves water but reduces carbon uptake and thus growth. The optimal stomatal aperture depends on water availability, light intensity, temperature, atmospheric carbon dioxide concentration, and the plant's current water status. Guard cells, the specialized cells that control stomatal opening, integrate information about all these factors, responding to hormones that signal the plant's hydration status, to blue light that indicates daytime conditions favorable for photosynthesis, to carbon dioxide levels inside the leaf, and to humidity and temperature. They make continuous adjustments to optimize the trade-off between carbon gain and water loss, solving what engineers would recognize as a multi-objective optimization problem in real time. What all these examples share is genuine choice. Plants select among alternative courses of action. They don't simply react in predetermined ways to stimuli. The response depends on context, on the plant's current state, its past history, and the range of environmental factors it's monitoring. Two plants of the same species in similar conditions might make different decisions based on subtle differences in their individual circumstances or developmental stage. This is the hallmark of intelligent behavior, context-dependent flexibility in the service of adaptive goals. Of course, we must be careful about how we use terms like decision and choice. Plants don't deliberate consciously. They don't weigh options in their mind's eye. The mechanisms are biochemical and biophysical, hormone gradients, gene expression cascades, ion fluxes across membranes, mechanical forces, electrical signals. But these mechanisms implement functions that, in animals, we would unhesitatingly describe as decision-making. The substrate is different, but the computation is analogous. This realization challenges us to reconsider what we mean by intelligence, cognition, and mind. These terms, so long associated exclusively with brains and nervous systems, might describe functional capabilities rather than specific structures. Intelligence could be understood as the capacity for adaptive, flexible behavior based on information processing and learning. Cognition could mean the mechanisms by which organisms acquire, store, and use information to guide behavior. Mind might refer to the integrated system of sensing, signaling, memory, and decision-making that allows an organism to navigate its environment successfully. Under these functional definitions, plants display intelligence, engage in cognition, and possess something we might call a mind, albeit one radically different in architecture and phenomenology from anything in the animal kingdom. The philosophical implications are profound. For most of Western history, we've drawn a sharp line between humans and animals based on rationality and language, and an even sharper line between animals and plants based on sentience and agency. Animals, we acknowledged, can feel and move and make choices. Plants, we insisted, are passive and automatic, alive but not aware, growing but not acting. The discovery of plant intelligence, if we accept that phrase, blurs these boundaries. It suggests that agency, the capacity to sense, evaluate, and act purposefully in the world, is not limited to beings with nervous systems, but is a more fundamental property of life itself. This doesn't mean plants are conscious in the way animals are. We have no reason to think plants have subjective experiences, qualia, the felt quality of what it's like to be that organism. Consciousness, if it exists in plants at all, would be so alien to our experience that we can barely conceive of it. But the absence of consciousness doesn't negate the reality of plant intelligence. Much of what animals do, including humans, occurs without conscious awareness. Our immune systems make complex decisions about which cells to attack. Our autonomic nervous systems regulate heart rate, breathing, and digestion through sophisticated feedback mechanisms. These processes are intelligent in the functional sense even though they're not conscious. Plant intelligence might be similar, sophisticated, adaptive, information-driven, but operating below the threshold of whatever gives rise to felt experience. Yet as we recognize plant intelligence, we must also recognize our responsibility toward these intelligent beings. If plants sense their environment, remember experiences, communicate with neighbors, and make strategic decisions, then they're not mere objects to be used without consideration but subjects with their own form of existence, their own way of navigating the world. This doesn't necessarily mean plants have rights in the same way animals might. Moral consideration should arguably track the capacity for suffering, and plants almost certainly don't suffer in any way comparable to animals. But it does mean we should approach plants with respect and curiosity, recognizing them as fellow travelers in the grand experiment of life, organisms that have solved the problems of existence in ways radically different from but no less sophisticated than our own. The ecological and agricultural implications of plant intelligence are enormous. Understanding how plants communicate, make decisions, and respond to their environment opens new possibilities for sustainable agriculture. Rather than treating crops as passive recipients of fertilizer and pesticides, we might work with their natural capabilities, using their communication networks to improve resilience, breeding varieties that learn and adapt more effectively, and designing agricultural systems that preserve the underground networks that allow plants to share information and resources. The discovery that mycorrhizal networks connect plants into cooperative communities suggests that soil health is about far more than chemistry. It's about maintaining the living networks that allow ecosystems to function as integrated wholes. Modern agricultural practices, particularly tilling and use of broad-spectrum fungicides, can disrupt these networks, potentially reducing the ability of crops to access nutrients efficiently and to mount coordinated responses to pests and diseases. Farming practices that preserve mycorrhizal networks, using no-till methods and minimizing fungicide use, might produce more resilient crops even if they're nominally less intensive. We're also beginning to explore the possibility of using plants as biosensors, detecting environmental conditions through changes in their electrical signals or chemical emissions. Plants already monitor their environment continuously with exquisite sensitivity. If we can learn to read their signals, they might tell us about soil contamination, water stress, pathogen presence, or nutrient deficiencies long before these problems become visible. This would allow earlier intervention and more targeted responses, reducing waste and environmental impact. On a broader level, understanding plant intelligence transforms how we think about ecosystems. An ecosystem is not just a collection of individual organisms competing and occasionally cooperating, but a network of relationships and information flows. Plants exchange carbon, nutrients, water, and chemical signals through underground networks. They communicate warnings through volatile compounds in the air. They shape soil chemistry, influence local climate through transpiration, provide structure for countless other organisms, and participate in biogeochemical cycles that regulate the entire planet. They do all this not as passive participants but as active agents, sensing their environment, making strategic choices, and influencing the conditions in which they and their neighbors live. The carbon sequestration potential of the Wood Wide Web is particularly striking. Recent research suggests that plant roots, together with their mycorrhizal fungal partners, transfer approximately 60 gigatons of carbon into soil each year, more than the entire carbon content of Earth's atmosphere and living vegetation combined. Much of this carbon is stored not in plant biomass, but in the soil itself, stabilized by complex interactions between organic matter and soil minerals mediated by fungi and bacteria. This root fungal microbial network represents one of the planet's major carbon sinks, and its disruption through deforestation, development, and intensive agriculture could have profound implications for climate change. Conversely, protecting and restoring these networks could be a powerful strategy for carbon sequestration. As we move toward a future that must grapple with climate change, biodiversity loss, and the need to feed a growing human population while preserving natural systems, the intelligence of plants offers both inspiration and practical guidance. Plants have been solving problems for hundreds of millions of years, adapting to environmental change, optimizing resource use, defending against threats, and cooperating with partners across kingdoms of life. They've created the oxygen we breathe, the soils we grow food in, and much of the beauty we cherish in natural landscapes. They've done this without brains, without centralized control, through distributed networks of sensing, signaling, and coordinated response. There are lessons here for how we might organize our own systems to be more resilient, adaptive, and sustainable. And there's something else, something harder to quantify but perhaps equally important. Recognizing plant intelligence invites us into a different relationship with the living world. Instead of seeing nature as a backdrop to human activity, a resource to be managed and exploited, we might see it as a community of intelligent beings, each with its own way of existing, its own evolutionary history, its own stake in the future. This doesn't require anthropomorphizing plants, projecting human qualities onto them, but rather biocentrizing ourselves, recognizing that we're one form of intelligence among many, participants in a living world far more sophisticated and interconnected than we've historically understood. In the quiet of this moment, as these ideas settle into your awareness, consider the plants that surround you in your daily life, the trees that line your street, the grass in parks, the houseplants on windowsills, the forests that still cover vast regions of our planet. They're not mere scenery but thinking beings in their own right, not thinking in the way you think, not conscious in any way you'd recognize, but nonetheless perceiving, remembering, communicating, deciding, acting with purpose and flexibility in pursuit of their survival and flourishing. They operate on timescales slower than ours, days and months and years where we experience seconds and minutes, but this makes their intelligence no less real. A tree that lives for centuries is processing information about thousands of seasons, integrating vast amounts of data, making strategic decisions about when and how to grow, where to send resources, how to interact with the forest around it, participating in conversations that began before you were born and will continue long after you're gone. There's a strange comfort in this recognition, a sense of being part of something larger and older and more patient than human civilization. Our species has existed for perhaps 300,000 years. Flowering plants have been refining their intelligence for over 100 million years. They've survived mass extinctions, ice ages, dramatic climate shifts. They've adapted to nearly every terrestrial environment on Earth, from tropical rainforests to Arctic tundra, from deserts to mountain tops. They've learned, in that deep evolutionary sense of learning, how to persist, how to find the cracks and opportunities in any environment, how to make life possible for themselves and for the countless organisms that depend on them. And they're still here, still growing, still solving problems, still communicating through their underground networks and airborne signals, still making the millions of small decisions that add up to the continued flourishing of life on Earth. In recognizing their intelligence, we don't diminish our own but rather find our proper place in the community of life, one voice in a conversation that includes all the sensing, remembering, deciding beings with whom we share this planet. As you rest now, settling into sleep, let your awareness drift to the plant life around you. If there's a tree visible from your window, imagine its roots extending deep into earth, connected through fungal threads to other trees in ways you can't see but that are no less real for being invisible. Picture the slow electrical signals moving through its phloem, coordinating responses across its vast body. Imagine its leaves measuring the precise quality of light, its roots foraging for nutrients, its cells storing memories of past seasons. The tree is doing all this right now, has been doing it all day, will continue through the night while you sleep and tomorrow while you wake, a constant patient presence engaged in its own form of life that requires no acknowledgement from you but continues nonetheless, ancient and ongoing. And if you'd like to continue exploring these mysteries with me, to journey into other quiet corners of science and nature, I invite you to subscribe, to join this community of curious minds seeking understanding and peace. For now, though, let these thoughts fade into the background, let the wonder remain but the details dissolve, and feel yourself sinking deeper into rest, held by the knowledge that intelligence takes forms beyond imagining, that mind is not confined to skulls but diffuses through the living world in ways we're only beginning to understand, and that we're surrounded, always, by the hidden awareness of nature, the silent consciousness of plants engaging with their world in every moment, a green intelligence that asks nothing of us but whose existence enriches our own, and with that thought, that peaceful recognition of our place in a thinking world, let yourself drift now, gently, into sleep, into dreams, into the deep rest that heals and restores, knowing that when you wake the world will still be here, still green, still growing, still thinking its slow plant thoughts, still carrying on the ancient conversation that is life itself. And in that continuity there is peace, there is wonder, there is the quiet joy of being part of something so much larger than ourselves. And now, rest, simply rest, and let sleep carry you away.